So now starting my presentation, my work is rosmarinic acid exhibits anti-cancer effect via MARC4 inhibition. So basically my lab is the drug design and discovery lab. We have three aspects: NGS and functional genomics, structure-based drug design, and protein folding and dynamics. We deal in these three aspects. Here today I'll be discussing for the structure-based drug design, and we have different kinases that we target. My current presentation, it will be focused on MARC4. So now coming to MARC4, basically what it is a 752 amino acid serine threonine kinase. It phosphorylates microtubule associated protein, that is tau, and thereby it plays a very important role in cell cycle progression. And overexpression of MARC4 is involved in cancer progression as well as Alzheimer's disease, because tau phosphorylation, hyperphosphorylation is involved in Alzheimer's disease. So now coming to our work, initially what we did is we expressed in purified MARC4. We can see a pure band of MARC4, and these all are the biophysical characterization of MARC4. This is UV visible spectroscopy, fluorescence, and CD. So we characterized our isolated purified protein. And then coming to the importance of MARC4, it is a druggable target, basically for Alzheimer's, for cancer, for diet-induced obesity, and for type 2 diabetes. So it is like a very important druggable target. And thus we selected this for our structure-based study. This study focuses on kinase inhibitor drugs, because now coming to our work, it's like because we are living in India, which is like heritagely very, we have different vast varieties of Ayurveda system earlier, and that's why our focus is on like the natural compounds, because they have a wide therapeutic potential. They represent biological diversity, and they are a part of our natural wealth. So now coming to the basic of our work, what we did is we carry out virtual high throughput screening. This is our target kinase, and this is the compounds library, say zinc database, which has millions of compounds. This is the flow chart through which we go. Initially, we filter the compounds through ADMET properties and physicochemical properties, then Penn's filter, which follows Lipinski rule, then molecular docking, then interaction analysis, and finally, we obtained our hit, the best hit. And then we subject it to MD simulation studies, and then further to biological studies. So biological evaluation, like it involves cell cytotoxicity assays, kinase specific assays, fluorescence, and other biophysical techniques. Now, in this study, what we did is we initially took millions of compounds. Out of those million compounds, we obtained rosmarinic acid as the best inhibitor based on the earlier filter which I showed, and based on the molecular docking analysis. We found that rosmarinic acid was interacting with MARC4 and inhibiting its activity optimally and to the best out of all the compounds. So we proceeded with rosmarinic acid and MARC4 at the atomistic level, like to see the interaction processes and all for that. Now, this is the docking study. What we found is these all are the binding pocket residues of MARC4. And what we found is that rosmarinic acid goes and interacts with all these functionally important residues of MARC4, and thereby inhibits the activity of MARC4. We can see the rosmarinic acid in the binding pocket, and these show the specific interactions like van der Waals hydrogen bonds and all. Now, coming to MD simulation studies, we see the atomistic insight through molecular dynamic simulations of the protein and ligand complex. Like we can see, we have different parameters like radius of gyration, hydrogen bonds, and RMSD and RMSF values. We can see the red one, it is for the complex, and the green one, it is for the native free protein. So what we found through all our studies for molecular dynamic simulations, that once rosmarinic acid goes and binds to MARC4, the stability of the complex increases. And similarly, we can see the average number of hydrogen bonds was around 5 for the protein ligand complex. So it denotes that the complex is stable throughout the molecular dynamic simulation period. Similarly, we can see in RMSF and PCA vector analysis, and this is the total energy analysis. All these studies suggest that once rosmarinic acid goes and binds to MARC4, it increases the stability of MARC4, and the complex is stable throughout the trajectory of molecular dynamic simulation. This is the PC analysis. Then coming to in vitro studies, we carried out fluorescence binding studies through fluorescence spectrophotometer, and we found that we took 3 different temperatures just to have an insight into the thermodynamic parameters of this binding process. And this is the Stern-Volmer plot, modified Stern-Volmer plot and Van 't Hoff plot. And we obtained the thermodynamic parameters, and that suggested the spontaneous nature of the binding, and also suggested binding constant obtained was of very high order. Like it suggested that the significant strength of the binding of this process. Then we proceeded to ITC, isothermal titration calorimetry. It is a very sensitive technique to study protein ligand interactions, and it suggested that rosmarinic acid binds in an exothermic manner. This is the pattern for exothermic reaction, and it showed significant binding affinity of this ligand with this protein. These were all the binding parameters that we obtained, thermodynamic parameters. Then we, after ascertaining the fact that rosmarinic acid binds to MARC4 and with a significant affinity, the next step was to analyze the functional analysis. Like if it is going and binding to MARC4, what is its effect on the functionality? Like the MARC4 is a kinase, so it's having a kinase inhibition assay. What we did is, this is for zero concentration, that is in the absence of rosmarinic acid, we took this as 100% the activity. As we increased the concentration of rosmarinic acid, we found that there was a corresponding decrease in the activity of MARC4, and its IC50 was found to be 6.2 micromolar, which is of a very good range, and it's very close to other known inhibitors of MARC4 that has been approved and that has been co-crystallized. Then we proceeded to tau phosphorylation assay to see that how it affects the tau phosphorylation, and we found similar results that validates our earlier results. Then we proceeded to apoptosis and cell proliferation studies, and it was found that for the normal cells, like there was no effect, no toxicity effect of rosmarinic acid, while the cancerous cells, there was a decrease, like it inhibits that. The similar results were obtained for FACS and annexin-5 staining. And then coming to the summary of our study was that we took rosmarinic acid out of millions compounds after filtering it through computational approaches. And then we carried out to in vitro studies and cell-based studies. And we found that rosmarinic acid is a potential inhibitor of MARC4 that can be implicated in treatment of MARC4 directed diseases like Alzheimer's and cancer. So these are the conclusions of my work. MARC4 is a potential drug target for cancer. Rosmarinic acid goes and binds to the active site of MARC4, thereby inhibiting its kinase activity. And this study certainly provides a rationale for therapeutic evaluation of rosmarinic acid and further, like its rosmarinic acid-based chemical synthesized inhibitors in MARC4. And this work was published in scientific reports. Now coming to my acknowledgements, these are all my supervisors and my colleagues whom I would like to acknowledge for this work. Thank you.